Hello, fellow cultivators of curiosity. I'm Natalie Kaiser, your guide through the captivating universe where seeds, genes, and everything in between converge. Whether you're out in the field, orchestrating a symphony of pollen in the nursery, or decoding secrets in the genetics lab, you've just stepped into the epicenter of scientific discovery. In today's episode, we will be talking to Dr. Will Rutter, a USDA vegetable nematologist and pathologist, who will share how he is delivering insights and genetic material to growers, as well as public and private breeding collaborators. So if you could tell us the story of how you navigated to your position today, and then specifically highlight options that you had along the way and why you made the specific choices that you did.
Okay. Yeah, I went to college at Knox College in Galesburg, Illinois. So it was very small, liberal arts college, and they didn't really have a specific plant-based degree. So I did kind of a mixture of biology and biochemistry. So I double majored in biology and biochemistry. And then when I got ready to go to grad school, I knew I wanted to go into genetics or molecular biology. So I kind of tossed out, applications to different graduate schools, and some of them were, medical based, more medically focused. But one was Iowa State University, and I went there and I interviewed with the faculty there. And I come from kind of a farming associated background. My grandfather was a farmer in southeast Kansas my whole life, and so I spent summers down there, kind of helping him on the farm every year. And so I think interviewing with the faculty there at Iowa State, which is a very agriculturally centered school, got me really excited and got me really engaged. So that was the main reason I think that I chose to go to Iowa State. And then I just happened to do one rotation in a plant nematology lab, and just happened to get into that lab, and the rest is kind of history. I went straight to PhD. I studied molecular plant nematode interaction. So I actually identified and nematode effector proteins that they secrete into the plant, and try to understand how those work and how those interact within the plant. And then after that, I knew I wanted to do research, so I kind of sent out feelers to different labs, and I got a call back from this crazy Russian guy, Edward Akunov, there at Kansas State University. So he was looking for somebody to study wheat stem rust interactions. So I went to his lab, and there I got a really good crash course in bioinformatics and wheat genetics, which is really complex. And I got to work on a whole new system, but still stay in plant microbe interactions and kind of apply what I had learned in my PhD there. And so that worked out really well. And I think I could have gone a lot of different directions, after that, having kind of the broad experience in plant microbe interactions. But actually, nematologists are in high demand right now. So anybody with a little bit of experience in nematology, really has a lot of opportunities right in the past few years. And one of those opportunities was to go to the USDA here in Charleston. And what really attracted me there is that I'd not only be able to work on the nematode side, but also kind of apply that to real world problems in vegetables. So that really appealed to me, that kind of translational research, bringing all the work I'd done on the molecular side and try to come up with solutions for growers. That was always really a big draw for me. So I've started there in February 2017, and I've just passed my 8th year here at USDA. So I've just been plugging away, looking at nematodes in different vegetable crops. Yeah.
And when you joined the USDA, was it clear to you as a younger scientist what were the expectations to succeed in that role, or is that something you've defined for yourself as you've grown over these 8 years?
I think the standards for success in ARS are laid out pretty clearly. You can you kind of have the option of depending on I was lucky enough to walk into an established program that had already been doing some working with different breeding programs for a while. So that was an obvious project to pick up and start moving forward. But it also has the added benefit is that I can kind of tailor things, and as long as I stay under my purview, which is nematodes and vegetable crops, I can kind of make the program my own, and I feel I kind of have since then. So it kind of pulled along some of the older projects and also kind of expanded into different areas of interest. I think have been very productive. So sweet potatoes, and I work on sweet potatoes, peppers, watermelon, and cucumber. So
That's a fun assortment of crops. Nice diversity.
Yeah. And they all have their little, it's very interesting working with, every time you come up with a new crop, each one has its own little foibles and intricacies that I've gotten to learn, and kind of each one takes a little bit different approach, I think. So, yeah.
You mentioned nematologists are in high demand right now. Are there any specific factors that are creating that vacuum? Or is it just the demographics of the way folks have come into the industry?
I think there's a number of factors. One is probably the phasing out of a lot of the most effective broad spectrum nematicides. So you think about methobromide was used for around 50 years. A lot of the organophosphates, that were really effective, that were used pretty widely by vegetable growers, just basically kill everything in the soil. But we're still had a high degree of environmental impacts. They were very effective nematicides. So a lot of times growers didn't have to worry about, nematode resistance because, they tear them pretty readily with just chemical applications. But now with the kind of the banning of many of those substances, a lot of nematode pests are coming back in a big way. And also with the, increased globalism over the past few decades, you get the movement of a lot of different plants, and with those nematode pests. So we've seen a lot of introduction of different nematode pests in different regions. So there's been a lot of high demand for anybody who has expertise in that area, but also the, there's not a lot of nematology programs, like specific traditional nematology programs in existence still. And a lot of the applied nematology expertise is kind of has fallen by the wayside, I think. So there's been a big push to get, genetics. There's a lot of geneticists out there. There's a lot of people with molecular biology understanding, but there's not a lot of people who could, effectively look at a bunch of different nematodes and tell you the genres or, even the orders that they're looking at, or what ones are a threat and which ones aren't. So it's a very specific skill set that there aren't a lot of people available who have. And I wouldn't even count myself as a very good nematologist, to be quite honest with you. I'm good enough, though, I guess, for my career. So I'm interested in learning more. So yeah.
Well, maybe in that vein, you are co-PI on the Find Me project. Could you tell us a little bit more about that project, focusing on understanding distribution and extent of this new pest or emerging
Yeah,
pest?
so the Find Me program is a specialty crop research initiative focusing on a specific species of root knot nematode, Meloidogyne enterolobii, sometimes called the guava root knot nematode. But this nematode is become a serious problem in the southeastern United States, mainly because it's a highly virulent nematode that's able to break many of the resistance genes that have been used in crops for many years. It's simply just those same genes that are effective against the endemic species of root knot nematode aren't effective against Meloidogyne enterolobii. And it's also gotten into several different nursery crops instead of specifically sweet potato that have kind of facilitated the movement of this nematode around our region and caused it to become even more of a problem. So our goal within the Find Me project is to try to define the distribution of this nematode, come up with solutions to help slow its spread and help manage or help growers manage this nematode in fields that already have it. But my main focus in that project is development of identification, development of new sources of resistance against Meloidogyne enterolobii in several different crops, and developing new methods to detect and survey for this nematode, because it's actually very closely related to many of the endemic species of root knot nematode. There's very few people who can look at this nematode even under a microscope and tell you which species is which. So it normally requires a molecular test to actually identify this nematode.
So a component of this is helping develop strategies for control. And that can sometimes be a sensitive topic with growers who have been growing in a certain operation for a long time and are comfortable in that routine. What is your strategy for making recommendations for new control procedures and protocols, and the education piece once you've gathered enough information to share results?
Yeah, we're doing that. We try to reach out to growers directly through field days. We've also developed different management and educational pamphlets to kind of reach out and try to connect with growers directly. I've had the opportunity to kind of work with some specific growers and home gardeners as well, just kind of trying to, they come with problems, and I kind of try to explain the situation of if they have enterolobii, and what they can do to try to manage it. It seems like most growers are interested in trying to, usually people don't come to us, unfortunately, before they have a problem. It's usually at the point where things are, when you have a nematode like this, it's usually when things are kind of crashing and you realize you have a serious problem. So they're usually interested in finding whatever solutions you can give them. But I think most of the management solutions we have now are usually changing up rotational schemes to try to reduce numbers in the field. So most growers, I think, I don't think it's in most cases a huge divergence from what they've done in the past. It's just a matter of making them understand, how different rotational schemes might cause increases or decreases in the nematode population, and let them know that there's probably no way they're going to get rid of this nematode. It's really just going to be a constant battle.
A mitigation strategy.
Exactly,
yeah.
a mitigation. Yeah, exactly.
Have you been surprised at all by your findings of the distribution and the relative velocity of the spread compared to other nematode populations you've study?
Yeah, I haven't. It's actually kind of rare to step into a position at the exact right time when things are, nematodes in general don't spread very fast. It's usually when they do spread, it's usually um, anthropogenic, human
Mhm.
based, and that's the case. And so it was really when I first started, we didn't know if we had it in the state. We'd heard rumors that it was in North Carolina, and I had never speciated it, so we were just kind of like looking around, seeing if um, we heard it was becoming a problem on sweet potato, so we specifically started looking on sweet potato. And once we founded the first instance of it, it just started popping up everywhere. We found it in the grocery stores. We found it at farmers markets. So it was just really surprising how easy it was to spread on sweet potatoes specifically. And sweet potatoes not the only. I don't want to vilify it. It's really, sweet potatoes have just kind of been the canary in the coal mine, if you will, because they're obligated to look at their roots. But there's another. I mean, this same nematode infects soybean, as well as most vegetable crops. So most crops are going to be infected by. It just happens that sweet potato, because it's a clonally propagated crop, that's moving around roots every year, that becomes an easy vector for the nematode. If one infected root gets into a field, and allows that nematode to establish that that field is infected permanently, usually. So, I think it was kind of surprising how prevalent, how much it was moving around on sweet potato. I'd say when we first looked, I could have easily walked down the street and found it, in the grocery. I knew which grocery stores had it,
Mhm.
and it was just kind of, and you think about how easy it would be if those same sweet potatoes made it out into a field. Well, it's probably infesting that field. So, I think it was kind of astounding in that sense, how prevalent it was. Yeah.
Are you seeing any reports of a synergistic effect between interlobii infestation and any root vascular diseases, any pathology, infections that are made worse by the prevalence of this nematode?
Yeah. For this nematode in particular, there are pretty well substantiated reports in guava, and so the reason it's referred to as the guava root knot nematode is that it first came to, I think, prominence in South America, Brazil, in the Brazilian guava industry, where they showed that it had some synergistic interaction with, I believe, some fusarium species, some fusarium root rots, where the two pathogens got together. You saw really drastic declines in guava stands. And there's been, similar reports for other species of root knot nematode with fusarium and cis nematode interacting with fusarium. Specifically, we think of soybean cis nematode and fusarium in soybean. So it's considered soybean a major factor in sudden death syndrome, soybean sudden death syndrome. But it's really hard to effectively demonstrate that kind of synergistic interaction. You can definitely imagine how, the nematodes moving into the root could also bring other pathogens into the root at the same time. So it's definitely feasible, but I don't think we've seen anything definitive showing us what I would call a synergistic interaction with enterolobii on it, anything in the US yet. But not to say it couldn't, it's not possible. Yeah.
It's not yet.
Yeah.
What about any promising results in terms of genetic resistance to enterolobii in any of the crop species that you have examined?
Yeah. I mean, there's really promising. Luckily, we have a really great germplasm repository in the USDA for sweet potato, and there's good news in sweet potato is that although it's a really efficient vector and all the commercial varieties are highly susceptible, we found a lot of breeding clones that have a high level of resistance. So about 20% of the PI lines, the plant introduction lines from the USDA collection, we found to have a high level of resistance to Meloidogyne enterolobii. So that, and we already have free breeding programs for sweet potato in the U.S. So there's one at NC State, there's one in LSU, and we have one major one here at the station as well, and, along with a few others. And I think all of them now, there's a big push to incorporate these different sources of resistance into their newest lines. They've all incorporated resistance into their breeding programs, and now it's just a push of trying to identify sweet potato lines that have the full horticultural package that people are looking for. And I know NCSU has developed a marker for their source of resistance in Jewel, and I think we just recently finished a genome-wide association study where we identified some markers associated with some different loci as well. So it's potential we have multiple sources of resistance against enterolobii, and hopefully in the not too distant future, we'll start seeing, horticulturally favorable lines with guava root knot nematode resistance. Yeah.
That's encouraging. It's not often the case that you have such a high percentage of resistance in your gene bank.
Yeah, that's definitely not the case we've found in pepper or in cucumber. So in pepper, we've screened over 400 PI lines, and we've come up with one line that what I would call moderate resistance, almost more like a tolerance. Nothing like the strong resistance we see in the endemic species. In cucumber, cucumber is highly susceptible to many root knot nematodes, and we don't see any resistance in that germplasm either. We do see we have found some wild watermelon that's Citrulus amorus that has a pretty high level of resistance to enterolobii, and so we're working with our watermelon breeders to try to move that into the cultivated species Lanatus. So that's a hopeful story as well. So yeah.
You mentioned screening. Are you able to use assays with high concordance to field conditions for all of your crops, or do you rely on infested fields for identification of tolerance?
Yeah. Most of our screening, because it's so labor intensive, really has to be done in the greenhouse. It's very, screening for resistance in the field. I won't say it's impossible, but it's very difficult, just because due to usually the spotty nature of root knot nematode infection. Like you'll have one really high, individual spots that are very hot, high populations, and other spots that have hardly anything at all. So you get a lot of false positives and potentially false negatives as well. When you do it in a field condition. So most of our initial assays are all conducted in the greenhouse under pretty as tight of conditions as we can to kind of make good comparisons between different plant genotypes. Yeah. But eventually, once we get those, towards the what I would not call a variety, but more a line that we think is ready to release, that's really when we're starting to bring it out into the field and kind of try to help gauge its horticultural traits a little bit better. Yeah.
On that larger field scale. Yeah.
Yeah. Yeah.
Well, maybe now stepping back a little bit from the specifics of this nematode project, I'm curious to know, how do you stay abreast of emerging threats to vegetable production? You have all of this intensive research going on that needs attention. How do you ensure that you have your ear to the soil or your eye on the landscape?
Yeah. Yeah. And, I think I do the best I can. You know, uh, I think trying to engage with growers, go to grower meetings, and see what's on the top of their mind is a big portion of that. I'm also lucky enough to have a lot of colleagues in academia, a lot of extension colleagues who undoubtedly do a lot better job of it than I do. So I get to hear what they're talking about and what they are focusing on as serious problems. Um, but I think those are two of the main avenues that I've taken to try to stay abreast of that and see what potentially needs to be addressed in the future. Yeah, yeah, and that's where we get back to the green fruit anthracnose project, which we're really excited about. Is a serious problem in pepper, and seems to be one that would really low hanging fruit, so to speak, or one that could easily be simultaneously addressed with nematode resistance as well. So that seemed like a something, an opportunity for us to kind of work with different pathologists to try to bring something to market that is, or to breeders that has multiple resistance traits. So I think anytime you can work with other pathologists to try to develop something with multiple resistance to multiple pathogens, that makes it all the more appealing to growers and breeders as well. So
When you have that one-stop shop solution.
exactly, yeah, exactly. If you can stack traits on top, favorable resistance traits on top of each other, it just makes the job of the breeder all that much easier in the future.
You mentioned the green fruit anthracnose project. Could you give us just a quick synopsis of that project and maybe how that collaboration came into being? Because that has folks from a lot of different functionalities involved.
Yeah, yeah. So green fruit anthracnose is a kind of a re-emerging pathogen. Anthracnose, there's many different anthracnose pathogens, but specifically this one is Colletotrium scovillei, and it has been causing some serious outbreaks both in the Northeast and the New York area, as well as the Southeast down here in the area of South Carolina. And so I really learned about it through our Clemson colleague Tony Kynath, who was had spoken about it. And because I was working on pepper, a small pepper breeding program here, he asked if I had any lines that they could test for this pathogen in the field. And so I got a chance to kind of collaborate with him on that field project, looking at the susceptibility of different pepper varieties to green fruit anthracnose, and found some really interesting differences for different chili varieties against this pathogen. And it just so happened that it coincided with some of the root knot nematode screening I was doing as well. So it was an opportunity to kind of work with Tony and develop some potential populations that could be useful to help map the resistance we were seeing. And he developed the collaboration with Sarah Pethybridge up at Cornell, and working with the growers up there in the banana pepper industry up there that were getting hit very hard by this pathogen. So that's kind of been a really great collaboration. We've got a little bit of funding to start working on that, hopefully. and hopefully be able to kind of identify some useful resistance traits that we could add to our breeding lines in the future. Yeah. So
I'm looking forward to hear how that shapes up.
yeah, I hope so too. Yeah.
So switching gears a little bit again, because you have such a diverse set of engagements. You're an author on a recent publication delivering haplotype resolved pepper genome assembly. Could you speak a little bit to how that collaboration came into being, and then walk us through what's the novelty of this approach, and what does it afford?
Yeah. So that was a really interesting collaboration with one of our ARS bioinformaticians, Amanda Hulskamp. She's one of our scientists here in the Southeast area who help on a bunch of different crops, specifically with genomics. And then we hired a postdoc, Emily DeLorean, who's since moved on to breeding with hazelnuts up in Minnesota, is my understanding. But that was kind of their brainchild. I kind of brought to them a problem when I first started. I really wanted to incorporate marker-assisted selection into my breeding program because, as I mentioned before, phenotyping for root knot nematode resistance is very labor intensive. Like a 100 plant screen, you're talking about 8 weeks and 120 man hours to screen just 100 plants. So it becomes very labor intensive and very limiting in what type of breeding you can do. So incorporating marker assisted selection could really bring some serious advantages. And although there are several root knot nematode resistance genes in pepper that are known, they all reside within a region within chromosome 9 of the pepper genome that is highly repetitive. And as soon as I started looking into the literature and trying to figure out how I could design markers against specific genes, it became abundantly clear that none of the publicly available genomes were correct, or we didn't know which one was correct at the time, if any of them. It was just so repetitive that this region, highly repetitive region with a bunch of resistance genes in it, just wasn't consistent amongst the different genomes that were available at the time. So the goal of the project was really to try to resolve the repetitiveness of this region. And so I was lucky to have 2 double haploid lines that were originally developed by a French group back in the 80s that had different nematode resistance genes within this region. And Amanda and Emily came up with the idea of actually, because pepper is such a huge genome, it's very expensive to create a very good assembly. We're talking about larger than the human genome, or near so. So instead of going after just choosing one and just going after one genome, we decided to take this approach of sequencing not the individual parents themselves, but their F1 hybrid. So we sequenced the F1 hybrid using PacBio long read sequencing, while at the same time we also sequenced the parents using short read Illumina, high density Illumina sequencing. And by doing this, we could take the more expensive PacBio derived reads and use the Illumina reads that from the parents to basically sort them into their proper bin. So that's why we refer to it as trio binning. We can take the haplotype resolved reads, PacBio reads, and sort them into which parental pool, and then use all of that data to assemble the individual genomes of the parents. So we're essentially getting, you know, 2 high-quality genomes for the price of 1. And it's it turned out to be a really powerful approach. You know, we got several, several of the chromosomes, were telomere to telomere assemblies. So it really turned out nicely, and we're really hoping we're to have a paper out pretty soon showing how much better the assemblies are within our region of interest than previously publicly available genomes were. So we're finding some really interesting data that's really kind of shining light on why it's been so hard to conduct marker systems, identify markers against these different genes. So we're hopeful that we'll be able to get that data out pretty soon. So, yeah.
That was my next question. What ramifications did it have for your mass program?
Yeah, Marcus. Yeah, so it's very tough. It's been very tough for some of these genes to find a consistent marker. We find I can get a marker for specific populations that will work, but then trying to apply that marker to a different population becomes very challenging because the marker just isn't acting the same. And it's what we're seeing is actually we're massive insertions and deletions. Like we found an insertion of over 1.5 megabases in length inserted within this region. Wow. With a bunch of genes in it. Yeah. So it becomes really complex, and you have to imagine when you have insertions that large, it's probably affecting recombination frequency in that region. So it's really quite a story, I think, and worthy of looking into and making people aware of. But I think many of the markers that have been published are work if you can get them to work on your specific populations, can work well. But they definitely have certain lines are massively different, specifically in this repetitive region. And so I think that's something that's worth looking out for different programs, depending on what your parental lineage looks like, I think. So
were there any specific pepper types that were particularly problematic for identifying a predictive marker that you're excited that this new information will enable markers in those pepper types?
Yeah, well, I can specifically speak to I don't work with a lot of different pepper varieties, but I can speak to specific genes, specifically ME1, which is a resistance gene that has been shown by our lab and other labs to be more durable than the other resistance genes that are available. But it's been very difficult to actually design markers against and actually use. And we're finding it's because of these large insertions in there that's really interfering with that with marker development. And so hopefully we'll be able to resolve that here in the near future.
Super exciting.
Yeah.
You mentioned in the beginning that one of the draws to a USDA RS position was the ability to do translational research and have those real world impacts. Do you have a story from your career about some research that delivered tangible results that you're excited about or making changes in the real world?
Yeah. So one of the things we're really part of the Find ME project was in addition to finding new sources of resistance, I was also part of the project that looking to develop new ways to really survey for this new nematode. Excuse me. And one of the problems is, as I said, this nematode is moving around on sweet potato, right? Seed sweet potatoes. And the problem is oftentimes, it's not obvious if there's a light infection, but individual potatoes can be infected with hundreds of females at once. They could be mixed populations with multiple species. that require a molecular test, and traditionally that would mean going in and actually excavating individual nematodes and then running molecular tests on them. But we kind of worked with a couple other labs to develop this new method to survey batches of sweet potato for the presence of low-level guava root knot nematode infection. So we affectionately call it the toilet brush approach. So we put it basically looks like a toilet brush on the end of a drill, and we take batches of sweet potato and actually skin the sweet potatoes. And because those nematodes are concentrated on the outer, right under the outer skin portion, we can then collect all of those skinnings together in like a homogenized sample. We can extract total DNA and then run species-specific PCR to actually detect for the presence of. So instead of looking at just 4 to 5 nematodes out of potentially hundreds, now we're looking at a community of nematodes to see if any of them are potentially our nematode of interest. So by doing that, we're able to detect M. enterolobii at really low levels, below the detection level of actual visual detection before visual symptoms are apparent. And we were able to show that we can detect as little as 2 eggs in a 10 mil sample of sweet potato skins. So that is being offered as a service now for growers looking to see if they have a problem with this nematode. We're hoping that we can extend it to make it actually applicable to clean seed programs, so they can monitor their seed and make sure they're not passing this nematode on to wherever those seed sweet potatoes are going. But I think that's has potential to have real impact, and give growers and regulators a new tool to kind of help slow the spread of enterolobii to new regions. Yeah.
And most importantly, it has a cool name. It has a catchy
exactly.
name.
The toilet brush method. Yeah.
Do you have any recommendations for one way that you would change the way that the next generation of plant scientists or plant pathologists are trained? If you could inject one element into the development of the next generation of scientists, what would that be?
I don't know if I'm qualified to dictate how new plant scientists should be educated, but I think any scientist, in biology, I think I would encourage them to kind of branch out, get experience in multiple different fields. In my experience, has been, I've really benefited from going from a strictly molecular lab. I got thrown into being the molecular guy in a bioinformatics lab, and then I got to move into a bunch of different vegetable systems and work with a bunch of different vegetable breeders. And each time I think I learned new things that I could apply in other systems. So I think it's that the idea of moving into different areas of research that you might not be comfortable in, but you can work with somebody who is comfortable in that system, is always a real benefit to any career, I think. Yeah. So
From your experience, about how many years or about how long did you find valuable to stay in each function before you were ready for a new challenge?
Oh man, yeah, I don't think I've ever stayed anywhere long enough to call myself, have to say that I've learned, everything there is to know in a specific area. But I know, I think staying a place at least a year is always good. I think it takes a year sometimes just to get your feet under you. And I don't, It all depends on the situation, but of course, I don't think you should be necessarily, anxious to get somewhere else. Hopefully, try to focus on learning and doing the best you can and with the job you have at the time. And I think it'll, most of the time it'll pan out for you and can reap the benefits of that. Yeah. So.
Well, staying in the philosophical esoteric vein, if you weren't a pathologist or a biologist, where do you think you would be?
Oh, if I wasn't a biologist, oh man, I think when I was a little kid, I've always wanted to be a biologist. I come from a line of biologists.
Yeah.
My dad was a biochemist. My grandfather was a biology teacher. So it's always been like, biologist, naturalist. But if I honestly, after working in a bioinformatics lab, I really did like computer science. I think that would have been awesome because things work every time. Like I don't know if that's really like, yeah, it's mind blowing for somebody coming from like a molecular lab where 6 month work of successful experiments came from like 5 years of unsuccessful experiments. So I think that's definitely would be a draw for me, I think, in another life. Yeah. So.
What about any crazy retirement plans? Any business you want to own?
Retirement plan? Oh man, I don't know. I think it would be cool to play with fruit breeding. I think fruit breeding is very cool. I don't know if I'd be any good at it, but I think it'd be fun to play with and just see the results.
Thank you so much for the time today. It was really a pleasure to chat with you.
Absolutely, anytime. I appreciate the invite.